Good morning, students. How are you all? So I hope all are fine. Good. So today's class, we are just recall that previous class concept, children. The last class we had studied virus, isn't it? What do you mean by virus? Very good. So that is one of the very ultra microscopic particles. So that is obligate intracellular parasite, isn't it? So that is the definition. We are framed that. So that's a definition for viruses, isn't it? Yes. What do you mean by virology, children? Study of viruses known as virology. Very good. The virus has two components, two essential components. What are they? Very good. Tell me. RNA and DNA content, isn't it? The nucleic acid either they present in a RNA or rRNA. Clear? Yes. Next one. What is that? The word meaning of virus? Poison. Very good. Okay. Yes. The next one we had learned that's the size and shape of virus. Do you remember that is the three shape of viruses, children? Yes, good. Tell me. Cuboid symmetry, helical symmetry, complex and atypical symmetry. Okay, so these are the three types of the symmetries, isn't it? So based on that you have to decode that's one on examples also. Clear? Yes, good, very good. The next one. What is that? Characteristic features of virus. The characteristic features of viruses means, so there is a living characteristic features and non-living characteristic features. Clear? This is a living and non-living characters. So here that is a living means some of the peculiar characters, some of the certain characteristic features of this viruses. Then non-living means some of the characters, isn't it? So can you able to say some of this one or two living characteristic features of viruses? Yes, first one, the ability have mutation, isn't it? Mutation means a sudden hereditable changes. Clear? Yes, good. The next one. They have nucleic acid and protein coat. Then the ability to multiply inside the host cell. Clear? So some of the living characters. Non-living? Very good. The absence of metabolic activities. They presence in a crystalline form. Clear? Main points only I am telling. Okay, then. Very good. So the absence of energy producing enzyme system. Understood, no? So these are the certain characteristic features of non-living organisms. Understood? That's the non-living characteristic features of viruses. Yes, good. Next one. Very good. Classification of virus. Clear? The classification, how many classes are there? Very good. 7 classes. So based on that 3 main characters based only, they are classifying. That's the classification of this viruses, isn't it? Then 7 classes and also some of the examples of the viruses there. You must prepare it. Okay, children? Yes. So there you have to be prepared. That's some of the one word. Who discovered that is the. That's very good. There's a classification. So who proposed in which year? These all the things also you must prepare it. Clear, ma? Yes. The next one is viral genome. Then the nature of this viral genome based. So that is very good. So they are giving in a certain conditions, isn't it? Then what do you mean by deoxyviruses and what do you mean by riboviruses? Riboviruses means what is the nucleic acid content is there? Ribovirus. Very good. RNA content. Understood? Then what is the nucleic acid content is present in a deoxyvirus? DNA content. Clear, children? Yes. Very good. So then there also there is based on that genome, the natural genome, that based they are dividing in a 4 categories. Do you remember that 4 categories, children? Yes. Double-stranded viruses with double-stranded RNA, single-stranded RNA, double-stranded DNA and single-stranded DNA. Isn't it correct? Yes, good. So there also you have to be quoted in some examples. Understood? Then we had learned there is one of the detailed study of the two viruses, isn't it? Examples of the two viruses. One is tobacco mosaic virus and another one is bacteriophage viruses. Clear, children? Tobacco mosaic viruses, from which plants obtained or discovered that tobacco mosaic virus? Very good. So there is a tobacco plant. Clear? So there also who discovered this is the tobacco mosaic virus, in which year? That's the things you must learn, children, for that one word based. And the next, very good. What are the symptoms appearing in one of the normal tobacco plant is there means, suppose this virus is entered to affecting that is a normal plant. So that plants, so they get it in, that is the tobacco mosaic diseases, isn't it? At that time, what are the symptoms to appearing in this is a tobacco mosaic viruses. Okay, so that points also you have to be prepared in that places. Understood, pa? Say yes or no, da? Yes, very good. The next one, structure of tobacco mosaic viruses. So already I taught that is the what about the structure. Want to learn about, want to prepare, there is some points regarding that's the tobacco mosaic virus. Clear? Yes. Next one, phage viruses, isn't it? So there is a phage viruses. So they comes under so many phage viruses. T2 phage, T4 phage and T6 phage. These are some of the names, some of the subtypes of this bacteriophage viruses. Okay. What do you mean by bacteriophage, children? Very good. Virus infecting the bacterium. So, okay, the bacterium, very good. So, the viruses infecting the bacterium is known as bacteriophage viruses. Clear? Mostly where are the places they occur bacteriophage viruses? Soil, vegetables, milk, isn't it? So, these are the areas mostly they found that is the very good, the bacteriophage viruses. Then we had completed that is the structure of bacteriophage, isn't it? The structure of bacteriophage, what are the structures are there, children? If we are making, this is the bacteriophage, so this like one structure means what are the items are there? Yes, very good. So there is the part is head, collar, very good, tail, basal plate. So these all the things are the presence in a very good. That's a bacteriophage, isn't it? Then whatever that content is presence in a headed region and tail part, what is the use? So these are the things we learned that we had completed in a previous class. Are you understood, children? Say yes or no? Are you clear the concept? Yes, very good. So today's class we are moving the next portion. The multiplication or the life cycle of a phage. Okay, the multiplication or replication or the life cycle of the phages. So here, based on that life cycles, okay, the phage, they comes under to categorizing in two types. One is lytic cycle and another one is lysogenic cycle. Lytic cycle means we are telling it that it's a virulent cycle, and lysogenic cycle means so that is called as avirulent. So that's a non-virulent type of cycle. Clear, children? So these are the through two types only the life cycle of the phages is going on. Clear? The first one we are going to prepare. This is the lytic cycle of this multiplication of this virus, or that is the what is the life cycle of this lytic. Clear? The first one, if we are making this is a lytic or virulent cycle. Okay. First one, if that, what do you mean by lytic cycle means? So all of you listen. The phage particles, already I said that is the phage particle. This is the phage particle, isn't it? This phage particles, so that is the DNAs are not integrated. They disintegrated with the bacterial chromosomes. That's the type is we are called as, the name is known as lytic cycles. Clear children? The lytic means, what do you mean by lytic? So there is that's a phage DNA is not disintegrated. I mean, so that is disintegrated to the phage DNA, or that is the bacterial chromosomes. Clear? So that is known as lytic cycle. So in this steps, so this type of this multiplication, this type of the lytic cycle, the phage, the full virions also releasing. This is the first multiplication, or that's the life cycle of phage particles. Understood children? This lytic cycle is also called as, the name is known as virulent cycle. Clear? Yes. Here, if we are looking, this is the lytic cycle, some of the steps involving this. Okay? So what are the steps involving in the lytic cycles? The first one is adsorption, penetration, synthesis, assembly and maturation, and release. Okay? So these are the some steps involved in a lytic cycle of this phage. Clear children? Let us learn that one by one. First one is adsorption. First one, the viruses, they making this contact between this is the two. Between this is the two means, first one, that's an interaction between the cell wall and the phage particles. Okay? The interaction between the two. One is cell wall and that is the phage particles. Clear? So this time the phage particles making the contact between these two tail fiber region of the phage particles. So here if we are looking, this is a one. This is the phage tails part. Okay, so this is the tail part. This tail part, first they recognizing, that is the recognition site of the bacterial cell. Clear? Then this tail fiber, so their presence in lipopolysaccharides. Okay? This lipopolysaccharide is present or found in the, that's a phage tail particles, the phage tail region. Okay? That is act as a receptor region. Clear? The first one, that's making contact between these two. One is, so this is the host cell, or that is the bacterial cell. This is the phage particle. These two are, they contact between these two. First one, the phage particles are, they recognition. So recognize, that is the particular, very good, the specific receptor region, receptor site. Okay, that region. So here, that's a tail region, their presence in a, find outing that, and then this is the tail part, their presence in a lipopolysaccharides molecule. That lipopolysaccharides molecule, that one is act as a receptor of this phage. Clear? Then next one, we are going to learn about, this is the two more questions in this part. What do you mean by lanting? Okay, what do you mean by lanting? First one, the process involving, that is the recognition to the phage to the bacterial cell is known as lanting. Okay, this is the bacterial, so this is the phage particles recognition to the bacterial cell wall. Okay, so that is known as, that processes we are called as, the name is known as lanting. The next one, pinning means. What do you mean by pinning? First one, this is the phage particle recognition of this bacterial cell wall. That process is known as, first step is lanting. Then pinning means, so here the contact is establishing these two, the tail fiber and this is the bacterial cell wall. So that one is, what happening means, the tail fibers are bent towards to fixing, that's the basal plate. Yesterday you saw that is the basal plate of this, the end regions of the phage particles. Clear? That one, bend it and fix into the cell wall surface of this bacterium. Clear, children? That processes the fixing of the basal plate to the surface of the bacterial cell, that is known as pinning. Clear, children? Are you understood now? What do you mean by landing and what do you mean by pinning? Pinning means just the bacterial phage particles, the fix, that's a basal plate region. The basal plate means the tail part is there, no? That one is fixed to the cell wall of this bacterial cell. Okay? The cell wall of the host cell. Okay? They are fixing that process, that's known as pinning. Clear, children? This is very, very important to 2-mark question, children. Either sometimes they will ask, that's a 3-mark also. What do you mean by landing and what do you mean by pinning? Clear? Yes. Next one, the second step, the involved, that's lytic cycle, penetrations. How to penetrating, this is the DNA of this phage particle inside this one. Okay, children. For first penetrating process that is involving, that is the mechanical and digestion of the cell wall regions. Clear? But here, if you want to digesting, this is the cell wall regions means their presence in the phage digesting enzymes. Okay? That phage digesting some enzyme is there. That enzyme name is known as lysosome. Understood, ma'am? For first one, the digesting enzymes or the presence in that phage particle. That enzyme name is known as lysosome. So, that lysosome, with the help of this lysosome, the phage particles, so there is a destroying, there is the cell wall region or digesting the cell wall regions of this host. Isn't it? Host means that's a bacterial cell. Understood, ma'am? Say yes or no? The next one, what do you mean by transcription? So here, after this is the contraction. Contraction means, so this is a phage particle and bacterial cell, isn't it? So this contraction is make it in this tube. What will happen means, this is the basal plate regions. Again they fix it in that is very tightly means. So that time through this is just opening of this basal plate region. The end region is there, that is a fixer. First we saw this is the pinning region, no? So that one. That's a basal plate is just opening. Through that region, okay, through this is the region, only DNA particles alone, the phage DNA particles alone enter inside the bacterial cell or enter inside the bacterial chromosomes. Understood, ma'am? Only they alone, that's the bacterial DNA is injected into the bacterial chromosome. That process is called transfection. That step is called transfection. Clear, children? Say yes or no? Yes. After this is the contraction of this phage particle and bacterial cell. So through this is the basal plate. Okay. That basal plate just they open at that way only. This is the phage DNA particles are entering into the bacterial chromosomes. Understood, ma'am? This process, this step is we are called only that injecting the phage particle, only injecting the DNA molecule to the host cell or that's a bacterial cell. That step is we are called as, the name is known as transfection. Understood, ma'am? Then what is the thing is remaining, children? Just you see here, this is the DNA particle. The phage DNA particle is injected into the bacterial cell. Is it clear? The next one, what's the thing is remaining? The phage particles. Very good. Just they living in the protein coat. Okay. This living outside of this empty protein coat, no, that is the name is we are called as, the name is known as ghost. Okay. The first one, what do you mean by transversion? You know that what's the processes happen there. The next one, what do you mean by host? For the phage particle, the DNA is injected into this the bacterial cell. After that, only the protein coat only leaving outside that cell. Okay, that empty protein coat is we are called as the name is known as ghost. Clear, children? Say yes or no, ma? Yes. The next one, the third step is synthesis. Okay, synthesis means productions. If once that's the entering of this phage DNA particles inside there is a bacterial chromosomes means immediately all the machinery items, all the metabolic items that takes over the custody of this, the phage DNA. Clear, children? Then immediately destroying of this productions of bacterial chromosomes and bacterial DNA replication and bacterial protein, everything is destroyed, and then all the machinery item, all the metabolic item takes over the controls of this which particle? Phage DNA particle. Understood, children? So this is the production. So here only the production, that synthesis of which particles? Only phage particles. Clear, children? Only they producing in a phage DNA particles and phage protein molecules, and also so there is a phage replication. These all the things only the productions of this. This is the third step of this synthesis. Understood, ma'am? This is very, very important one of this 5 mark question, children. So all of you listen carefully. The next one is the fourth steps. Okay, the fourth step is assembly and maturation. Understood? So what do you mean by assembly and maturation? Already this step, what happened means, this is the phage particles involved the producing of this all the phage items. Understood, ma'am? The phage item means, so there is a protein coat, tail fiber, isn't it? Collar region. So there is a tubular core like structure, that one. Then DNA of the phage particles. Everything is individual, the parts wise, that is synthesized. Okay. The next one, what do you mean by maturation means? The assembling of phage particles. Okay, the process of assembling of the phage particles, that process is known as maturation. Understood, children? So there is the process of assembling the phage particle, that's the processes we are called as, the name is known as maturation. If we are looking, this is the on 20 minutes time, after that's a 20 minutes time, around, so here there is the multiplication. So there is the 300 phage particles of the assembling in this is the area. Clear, children? Within after that's a 20 minutes time, around that's a 300 phage particles. The phage young ones are they produces, they are assembling in this area. Clear, children? But what do you mean by maturation? And then how many of the number of the phage particles are producer or assembling in this area? Clear? Then finally, that's the last step is the releasing. But how to releasing means, so here inside that's the bacterial cell, the more number of this phage particles are accumulated. Okay. Then what will happen in children? So here there is a no space. Okay. So the more numbers are occupied means, so this one, that's the phage particles are lysis of this bacterial cell wall, that's a new phages are coming out. Understood, ma'am? So this is a lysis means the breakdown of this cell wall region from this bacterial cell wall region. The new phage particles are releasing one by one. Understood, ma'am? So this is the last steps comes to, that is a lytic cycle of this life phage particle. So that's a release. Understood, children? Say yes or no? So this is one of the steps involving in a lytic and that's a virulent cycles of this multiplication of this phage particles. Clear? So this is one of the very important 5-mark question. But you have to be no need to prepare the now. This is the 5-mark pattern, children. Just you want to prepare, that is the some 2-mark and 3-mark and 1 word based. Okay, children. So just you mark it, children. First one, this is the first steps. So here you have to be prepared. That's a 2 question. One is landing and another one is pinning. Clear? Then second one, the penetration, that steps. So you should prepare transcription and another one is ghost. Clear, ma? The next one, so that's the 4th steps. So here you have to be prepared, that is the maturation. Understood, children? Say yes or no? So this is that this is the 3-mark and a 2-mark questions only. You must prepare in this area. Then first one, what do you mean by lytic cycle? That's the cycle also is to prepare it. Clear? Next one, we are moving with the second type of this. Okay, so that is the second life cycle of this phages, lysogenic cycle, or that is the avirulent cycle. Clear? First one, what do you mean by lysogenic cycle means? All of you listen. Lysogenic cycle means the DNA particles, so this is the phage DNA particles enter into the bacterial chromosome. The integrated form of this bacterial chromosomes, okay, that's the integrated form of this bacterial chromosomes, that is known as lysogenic cycle. Not that is the independent form of the phage particle is developing, children. Only this is the integrated form of this. So this is the phage DNA, this is the bacterial chromosome. So these two are integrated forms only. They multiply, children. That is the process. That is the life cycle is known as lysogenic cycle. Are you clear, children? Say yes or no? Yes. Next one. What do you mean by propage? Maximum the phage DNA molecules. Okay, this is the phage DNA molecules. First, that one is entering inside. This is the linear and become, so enter inside the host cell. They becomes to a circular N shape. Clear? Then this circular shape of this phage DNA, that is integrated with the bacterial chromosomes. Okay, this integrated DNA of the phage particles to the bacterium, that is known as, we are called as, the term is known as propage. But what do you mean by propage? Propage means, so that is the first the steps. So that is the phage DNA particle is entering in a linear, isn't it? So then after that, so they make it in a circular form. Then, so this DNA particle of this phage, that is integrated with the bacterial chromosomes, right? This integrated form of this phage particles to the bacterium, that is called as, okay, the integrated form of this phage DNA is known as, what is that name, children? Propage. Understood, ma'am? This is very important for, that is the one of the 3-mark question. What do you mean by propage? Then after multiplication, so here there is some of this receptor proteins are there, children. That is act as a multiplication process of this lysogenic cycle. Then, so whenever that is a cells are dividing, so that time the integrated form of this, so this is a propage, isn't it? This propage wise only, there is a multiplications, that's a phages particles are going on. Understood, children? Say yes or no? This is the second type of this, the second multiplications of this phage particles. Understood? Then today I am going to give, this is one of this work, children. These 2 diagrams. Okay, so all should please make it, draw it in your classwork note, children. Before this is the interactive section. Okay, before you are come to attending the interactive section, you must draw this diagram. Already the last class I given, that's a 2 diagram. One is the structure of tobacco mosaic virus and another one is very good. So that is the phage particle, the structure of bacteriophage, isn't it? Then also today's class also I am giving these two diagrams. So these two diagram, life cycles of the phage was also you have to draw it in your classwork note. Okay children? Yes. Thank you students. Thank you.